Peace be upon you and welcome back. In a new video we will talk about a new topic which is the complementary to the last topic which was MHC. This topic is antigen processing and presentation. These are our learning objectives, so providing knowledge about endogenous and exogenous antigen processing and their mechanism, the concept of cross-presenting of exogenous antigens and presentation of non-peptide antigens. Before we start, recall that in the last video, which was about MHC, about the last slides, we talked about the evidence for the antigen processing. So we learned that an antigen presenting cell needs an antigen to be processed before it can activate a T helper cell. So how did we know that? Some scientists did some experiments. For example, they did 3 experiments. The first experiment, they fixed the cell, rendering it metabolically inactive with a paraformaldehyde before exposure to antigen. Then the cell couldn't activate any T cell. In second experiment, they incubated both antigen and the antigen presenting cell. After that, they fixed the cell. The cell could activate T helper cells. In the last experiment, they fixed the cell with a glutaraldehyde, which is similar to paraformaldehyde, rendering the cell inactive in metabolic states. But what did they do? They processed the antigen in vitro, I mean manually. So before, or they gave the antigen presenting cell pre-digested antigenic peptides. Recall that before exposure to antigen, the cell was rendering inactive. But despite that, it could activate the helper cells. So from these insights, we will learn that antigen presentation or antigen processing is an inevitable process before antigen presenting cells could interact with a T helper cell. There is another topic. We will see some evidences about different routes of antigen presentation. So experiments done by 2 scientists, L.A. Morrison and T.J. Breichel, revealed that the antigenic peptides presented by class 1 and class 2 MHC molecules follow different routes. How did they know that? They used a set of T-cell clones specific for a known influenza virus antigen. Some T-cells recognized and presented the antigen on class 1 MHC molecule, and some others recognized the same antigen presented on MHC class 2. So hence, they concluded that class 1 presentation requires internal synthesis of virus proteins, as shown by a requirement that the target cell to be infected by a live virus. The inhibition of class 1 presentation observed when protein synthesis was blocked by some inhibitor drugs such as imitin. So this blocks the protein synthesis, internal protein synthesis. This is how they knew that class 1 presentation requires newly synthesized antigens or peptides. This is dictates, or it shows as endogenous antigen processing, as we see here. should be processed. For example, this is a newly produced antigen inside the cell, processed, the proteosomes, and so on. The second conclusion was class 2 presentation occur with either live or replication incompetent virus. So protein synthesis inhibitors had no effect, indicating that new protein synthesis is not necessary for class 2 molecule or MHC class 2 presentation, but instead it requires endocytic processing. So it requires to capture an antigen either with a phagocytosis or receptor-mediated endocytosis. So captures the antigen in some kind of vesicle or phagosomes, then exposing it to an acidic environment until it is gradually processed with high acid and proteases. So then it engages with a vesicle containing MHC class 2, then it's whole on it and it's expressed to the surface. Some drugs, such as chloroquine, which blocks endocytic processing within the cell, or they intervene the pH process of these phagosomes, they can block expression of MHC class 2. So now we know that, or we saw some evidences about different routes of antigen presentation. Now we will talk about endogenous pathway of antigen presentation. So, as we know, in eukaryotic cells, protein levels are very carefully regulated. Every protein is a subject of a continuous turnover and it is degraded at the rate which is generally defined by its half-life. So, I mean, every protein has its half-life. Some proteins have a shorter half-life, some have a longer half-life, so it depends on the protein. So whenever its half-life ends, it should undergo degradation. Or, for example, denaturated, misfolded, or abnormal proteins, also they should be degraded. Or defective ribosomal products are another class of compounds that should undergo degradation. The average half-life for cellular proteins is about, some of them are about 2 days, but some of them may be degraded in about 10 minutes. So the consequence of steady turnover of both normal and defective proteins is a constant deluge of degradation products within a cell. Most of these products will be degraded to their constituent amino acids so as to be recycled and used to produce new proteins, but some of them are not degraded but otherwise are selected to be expressed on MHC class 1. So I mean they are samples. The cell samples some of the proteins. Until now it is not known how a cell selects some proteins or some peptides to be expressed as a sampling of the level of protein production, or how much is the level of abnormal or normal proteins, and how a cell can select them, it is not known. So what is the benefit of this process? So the cell, for example, can check itself whether it produces normal proteins or not. For example, when the age of the cell gets near to the end, it gets senescent, the level of abnormal or misfolded proteins increase. So it gets close to its death, so it should go under apoptosis. And another new young cell replace it. There is other example. For example, in case of cancer cells, they produce some proteins which are not normally produced there, or produced but in very low level in normal state. But in case of cancer, they are much more produced. Or in case virus infected cells, some proteins are produced which don't belong to the cell. So for this process, the cell should sample itself and show these proteins on its cell membrane to immunocytes, whether the cell is normal or it is not normal. If the cell is not normal, it may induce or it may elicit immune response. This is the benefit of this process. So the first process in endogenous pathway is peptide generation by protease complexes called proteasomes. So intracellular proteins are degraded into short peptides by a cytosolic proteolytic system present in all cells. It's called proteasome. They are composed of two parts, a 20S base and a 19S regulator compound, which is bound to a site or to the base component. So the large 20S is composed of 14 subunits I'm talking about here. Let's give me a short time. Let's annotate it. Spotlight. Yes, it's composed of 14 subunits arrayed in a barrel-like structure of symmetrical rings. Many proteins target for proteolysis when they are tagged with this protein complex. Also, it's called ubiquitin. So whenever a protein is tagged with this one, it signals the proteasome to degrade it. So this ubiquitin protein conjugates into the proteasome complex through here. So it goes through the center, and the proteasome complex cleaves peptide bonds in an ATP dependent process. So it requires ATP to break the peptide bonds between the amino acids. And this process happens in the middle of this proteasome. This proteasome exists in every cell. This process also happens in every cell. So for example, when a protein is misfolded, it is abnormal, or a ribosome defective product, it occurs in all cells. But another version of much efficient proteasome is called immunoproteasome, exists in activated antigen presenting cells. So it differs from the normal proteasome by having these parts, which are B1I, B5I, and B2I. It is coded by some genes which are located near of LMP2 and LMP7 genes that encode replacement catalytic protein subunits that convert standard proteasome into an immunoproteasome. So it increases the production of peptides that bind efficiently to class 1 MHC molecules. Also, notice that these immunoproteasomes are especially induced by interferon gamma and tumor necrosis alpha. So that's why I meant they exist in activated antigen presenting cells. So the immunoproteasome, what is the difference between this and this in terms of efficiency or function. So the proteasome, the immunoproteasome, turns over more rapidly than a standard proteasome, possibly because the increased level of protein degradation in its presence may have consequence. So, for example, it is possible that some cases of autoimmunity results from increased processing of self-proteins, especially these cells with high number of these immunoproteasomes. So in terms of function, they differ in efficiency and antigen processing. They do it much efficiently than the standard proteasomes. Now we know that when a protein is produced inside the cell, how it is produced is tied with a ubiquitin, go through the proteasome, is degraded with much smaller peptides. So in next slide, we will see peptide transport from the cytosol to rough endoplasmic reticulum. Actually, insights from this topic came from some studies on a cell line called RMAS cell line, which was defective in some point of antigen transport or peptide transport from the cytosol to rough endoplasmic reticulum. But how did they know that? Actually, these defective cell lines could only express about 5% of normal level of class 1 MHC molecules. So despite they were producing normal level of class 1 alpha chain and beta 2 microglobulin. So a clue to the mutation came from Townsend and his colleagues that they feed these cells a pre-digested peptides and the level of class 1 MHC molecules on the membrane returned to normal. And they reasoned that, or they concluded from this experiment that peptides may be required to stabilize the interaction between both the alpha chain and beta 2 microglobulin. The basic compounds of MHC class 1. So they said that there could be a defective in the peptide transport system. Subsequent experiments showed that the defect in the RMSS cell line occurs in the protein that transports peptides from cytosol to rough endoplasmic reticulum, where the class 1 MHC molecules are sensitized. When these cells transfected with a normal or a functional gene encoding the transported protein, the cell began to return to normal. I mean, it produced the normal levels of class 1 MHC molecules on its cell membrane, and the transported protein was designed as TAP. It means transporter associated with antigen processing. It's a membrane spanning protein. So it has two domains. One of the domain protrudes to the lumen of rough endoplasmic reticulum, and other domain have a ATP binding cassette. It is also a heterodimer. Okay, it's composed of two parts, TAP1 and TAP2, which is faced to the cytosol. These TAP proteins exist in many cells which mediate ATP-dependent transport of amino acids, sugars, ions, and peptides. So peptides which were generated in the cytosol in a previous slide by the proteasome complex, they are translocated into rough endoplasmic reticulum by TAP, by a process that requires ATP hydrolysis. So TAP what is the characteristics of the peptides that are transported by TAP? So TAP proteins has affinity for peptides containing about 8 to 16 amino acids. About longer peptides, when they enter the raffinoplasmic reticulum, actually they are trimmed by another protein called ERAP. Okay, ERAP stands for endoplasmic reticulum aminopeptidase. In addition, it selects the peptides which are rich in hydrophobic or basic carboxyl terminal amino acids. If you remember, these were very useful for anchoring the peptide in the binding groove of MHC class 1. Thus, TAB is optimized to transport peptides that are most likely to interact with class 1 MHC molecules. TAP1 and TAP2 genes are located within the class 2 MHC region adjacent to LMP2 and LMP7, and also there is different allelic forms in the population. Defects in this or defects in the genes which encode these TAP proteins lead to disease syndrome that has aspects of both immunodeficiency and autoimmunity, which is called lymphocyte bear syndrome. Now we learned that how the peptides are generated in the cytosol and how it is transported to the place where MHC class 1 exists, I mean raffinoplasmic reticulum. Let's see in next slide. Chaperone-aided peptide assembly with MHC class 1 molecule. Like other proteins destined for plasma membrane, the alpha chain and beta 2 microglobulin components of class 1 molecules are sensitized on ribosomes on the rough endoplasmic reticulum. So assembly of these components to a stable form requires the presence of a peptide in the binding groove. So the assembly process involves several steps and include participation of molecular chaperones that facilitate the folding of the polypeptides. So the first molecular chaperone involved in the class 1 MHC molecule assembly is calnexin, as we see here, a resident membrane protein of endoplasmic reticulum. ERP57, a protein with enzymatic activity, and calnexin associate with free class 1 alpha chain and promote its folding, as we see here. When beta 2 microglobulins came around and bind to the alpha chain, calnexin is released, and also the complex is associated with the other proteins are called calreticulin, so we see here, and tapacin. Let's see why does this happen actually. That this tapacin promotes, or it is associated with the TAP protein, and it makes the distance between the TAP protein and this complex much more closer. As we know, the TAP protein aids in peptide transport to the lumen of endoplasmic reticulum. So it brings the TAP into a close proximity. Exoproteases in the endoplasmic reticulum will act on peptides which are not associated with class 1 MHC molecules. So one amino peptide is ERAP1. So it removes, what does it do is it removes the amino terminal residues from peptides to achieve optimum class 1 peptide binding group. It has a very little affinity for peptides shorter than 8 amino acids. So as a consequence of productive peptide binding, it means when a peptide exported or imported to, actually it's import because it's imported to lumen of endoplasmic reticulum, and it could bind in a preferential manner to class 1 MHC molecule. It causes the release of the previous compounds which were bound to class 1 MHC molecule, which were calreticulin and ERP57. So now the class 1 MHC molecules exit from rough endoplasmic reticulum and proceed to the cell surface via Golgi complex. Now, we learned that how protein is converted to peptides, or it is processed to peptides, how it is transported from the cytosol to rough endoplasmic reticulum, and how it is bound to class 1 MHC molecule by aid of these molecular chaperones. Now we finished class 1 MHC, we will jump to exogenous pathway. So about the exogenous pathway, as you know, antigen presenting cells can internalize particulate materials by simple phagocytosis, where the material is engulfed by pseudopods on the cell membrane, or by receptor-mediated endocytosis when the compound fast or the substance fast attached with the specific surface receptors. Macrophage and dendritic cell internalize antigen by both process, and most other antigen presenting cells, whether it is professional or non-professional, demonstrate little phagocytic activity and therefore typically internalize exogenous antigen only by endocytosis. So receptor-mediated endocytosis or pinocytosis. For example, B cells, if we suppose it is an antibody, so the antigen attached to the specific antigenic receptor, and so once an antigen is internalized, it is degraded to shorter peptides within compartments of endocytic processing pathway. The endocytic antigen processing pathway, it involves several increasingly acidic compartments, including early endosome, which the pH is about 6 to 6.5, late endosome and the endolysosome, and finally the lysosome, which the pH is about 4.5. Internalized antigens progresses through these compartments, encountering both the pH and the hydrolytic enzymes. So it is cut into smaller peptides in a successive manner until it meets a specific type of endosome, or it's called late endosome, which is a unique feature of antigen presenting cells, have this kind of endosome which possesses MHC class 2 containing compartment in which final degradation and peptide loading into the molecule occurs. Within these compartments of endocytic pathway, the antigen actually it is degraded to oligopeptides, about 13 to 18 residues that met up with the and bind with class 2 in the late endosome. So it is opposite of class 1 processing pathway, which they were going to trim antigens to accommodate class 1, which were about actually 8 to 13, or the optimal was about 9 amino acids. Because of hydrolytic enzymes are optimally active under acidic conditions, antigen presenting processing or antigen processing can be inhibited by intervening chemical agents such as chloroquine again, as well as lipoptin, which the chloroquine increases the pH of these compartments. The later inhibits proteases. So now we learned how an exogenous antigen is processed until now. So let's see what later happened, or what actually happened in other processes to MHC class 2 before it acquires the antigen, or before it joins the compartment which contain an antigen. Let's see. Since antigen-presenting cells express both class 1 and class 2 MHC molecules, some mechanism must exist to prevent the association between MHC class 2 molecule with the antigenic peptides destined for class 1, because as you know, when MHC class 2 molecules are synthesized within endoplasmic reticulum, these class 2 alpha and beta chain associate with a could associate with the antigen which are destined for class 1, as the process we learned in the previous slides during class 1 MHC and peptide processing. So what happened here, or what aids in this process, is a protein called invariant chain or CD74. This chain actually, it's a non-MHC coded protein, interacts with class 2 peptide binding groove and prevent any endogenously derived peptide from binding to it. The invariant chain has some important functions. For example, it prevents, as we talked about, it prevents the antigen binding to the alpha and beta, or the peptide binding groove of class 2. It's also a reason for its assembly. One other important function is the variant chain contains sorting signals in its cytoplasmic tape that direct the transport of class 2 MHC molecule from Golgi network to endocytic compartment. In experiments, when the invariant chain was defective, the class 2 MHC molecule remained primarily in the endoplasmic reticulum, and it couldn't pass through the cis Golgi. So this is how they reveal it is important for its assembly and its transport to the endocytic compartments. When this complex, the invariant chain associated with the class 2 MHC molecules, are transported through some compartments to the late endosome, by exposing to the acidic pH, it is successively trimmed, but a small part of it still remain in the peptide binding groove. It's called CLIP. So it stands for class 2 associated invariant chain peptide. It remains bind to the peptide binding groove, so as to prevent, also it's to prevent the association with any antigenic peptides, which are, for example, not distinct from, or they are not generated in the class 2 for class 2 MHC molecule. This is how actually these are transported to the late endosome when the antigenic peptide exists. There is another important notes, which are HLA-DO and HLA-DM. So let's see what are they and what's their function. About HLA-DM is a non-classical MHC class II molecules. It's required to catalyze the exchange of CLEAP with an antigenic peptide. So the DM alpha and beta genes are located near TAP and LMP genes in the MHC complex. Like other class II MHC molecules, it is a heterodimer, so in the structure it looks like class II. However, unlike class II molecule, is relatively non-polymorphic and it is not normally expressed on the cell membrane, but it's predominantly found in the endosomal compartment. So HLA-DM has been found to associate with MHC class II beta chain, and function is editing or removing CLEAP or some peptides that do not associate with class II in a preferential manner. But when, for example, a peptide binds to class II and makes a strong bond, so it is harder for HLA-DM to displace it with another peptide. So this becomes the end result of the MHC class II bound to an antigen, which later is transported to the cell surface. There is another non-classical MHC molecules called HLA-DO. It's also relatively non-polymorphic. Unlike other class MHC molecules, the expression is not induced by interferon gamma. HLA-DO has been found to act as a negative regulator of antigen binding, so it modulates the function of HLA-DM and changing the repertoire of peptides that preferentially bind to classical MHC class II. So when in cells that express both DO and DM, these two molecules associate strongly with each other through endoplasmic reticulum and maintain this interaction until all the way to the endosomal compartment. So actually about HLA-DO, originally it was observed in B cell and thymus, but in later it's also found in dendritic cells, where it is thought to play a role in maintenance of self-tolerance. Actually, in a study done by two scientists, actually one scientist with his colleagues called L.K. Denzin, by using diabetes-prone mice engineered to express HLA-DO, dendritic cells are known to be essential for establishment of self-tolerance. They also present autoantigens to self-reactive T cells. So reactive T cells develop in this non-obese diabetic animal, and this ultimately destroy pancreatic B cells and causing diabetes. So in this study, actually, development of diabetes was blocked by the presence of HLA-DO transgene in dendritic cells. This another work suggests that normal DO expression may play an important role in modulating diabetes mellitus behavior to ensure the presentation of self-peptide repertoire that encourage the tolerance of self-antigens. Interestingly, in wild-type mice and humans, DO expression is downregulated following dendritic cell activation by an antigen, releasing HLA-DM to carry out its normal function of encouraging the presentation of a diverse array of peptides that stimulate these antigen-presenting cells. Now we join the end of the slide. So now we've learned how an antigen in an exogenous pathway is being generated, bound at MHC class 2, and how MHC class 2 is transported to late endosome and bind with an antigen, and the function is modulated by non-classical MHC molecules, HLA-DO and HLA-DM. And this is the summary on both classes. This is for class 1, ERP, ERP57, tapacin, calreticulin. This is for class 2 actually, and let's go here until the end. Let's get back to, sorry, adjusted, okay. So there is another very important concept, which is cross-presentation of exogenous antigens. So it means when an exogenous antigen is acquired by an antigen-presenting cells, it's been diverted to be presented on MHC class 1 so as to stimulate a naive CD8 T cell. And why that? And why does this need to be to happen? Actually, when, for example, an antigen-presenting cell internalizes and processes an antigen to MHC class 2, it also expresses a co-stimulatory signal to stimulate CD4 T cells. So in this fashion, it can stimulate T helper cells or CD4 T cells. But how about other cells? I mean, except for antigen-presenting cells, about other cells, for example, when a somatic cell, a body cell is infected, for example, with a virus, despite this expression of the antigen on class 1, actually it can't elicit immune response, or it can't stimulate antigen-specific CD8 T cells because it doesn't express co-stimulatory signal. So how or what is the compensation for this if a cell is infected and can't stimulate CD8 T cells? So, or there is another question. And for example, for one process or for the previous question, it could lie in, for example, an antigen presenting cell should be infected with the same pathogen, or if a professional antigen presenting cell is not infected by an intracellular pathogen. So how does the cell present pathogens, for example, as a virus that have been engulfed from extracellular source in ways that it will activate the needed CTL response? Actually, these answers lie in cross-presentation. It means presentation of exogenous antigen and divert it into endogenous pathway. And why that? To activate antigen-specific CD8 T cells, as the antigen-presenting cell will also express co-stimulatory molecule to induce CT8 cytotoxic T cells to respond to the infection. And the most potent cross-presenting cell is a dendritic cell, which appears to reside in secondary lymphoid organs. So it receives antigen from extracellularly from either migrating antigen-presenting cell or from dying infected cells. There is also a few other cell types which also could cross-present, such as B cells, macrophages, and dendritic cell, but till now, there is no in vivo evidence for this. Oh, sorry, before we go to there, let's see if we have a go or if you have forgotten anything. No. We'll go to the mechanism of cross-presentation. At first, there was two hypotheses. So one was saying cross-presenting cells possess a special antigen processing machinery that allows loading of exogenous antigens onto class 1 molecules. The second was saying there is a specialized endocytose machinery that can send internalized antigen to an organelle, for example, phagosome or early endosome, where peptides from those antigens are loaded onto class 1 molecules. So let's see which is actually these two proposed mechanisms are not mutually exclusive and evidence for both exist, but actually in early 1990s, it was shown that bead conjugated antigens captured by cross-presenting dendritic cells, they could reach cytosol of the cell. So later, it was discovered that exogenously added cytochrome C, which causes programmed cell death when it is exposed to cytosol, it could induce cross-presenting dendritic cell to undergo apoptosis. So it is now proposed that retrotranslocation or movement of endocytosed protein out of endocytic compartment and into the cytosol, it occurs via the TAP protein we talked previously. So, however, TAP, also there is some TAP independent process. So it means that there could be multiple processes, but the export or the antigen from the endocytic compartment to the cytosol by TAP, it could be the most prominent pathway. So, for example, this is a virus infected cell. This is viral antigens. Also, we suppose that proteins are engulfed or picked up by antigen presenting cells. So it presents both viral and self antigens, and it will show it or it will show it on class 1 to CD8 T cells, for example. So let's see, it causes the sensitization or activation of these T cells or antigen-specific T cells because of having co-stimulatory signal. And what is the advantage of this process actually? When an antigen-presenting cell capture virus from extracellular environment or from, for example, a dying cell or an infected cell, it activates specific CD8 T cells so as to limit or inhibit further spread of the infection. So actually, there is some questions. So if the pathway is so important, why don't all antigen presenting cells cross present? So why not all antigen presenting cells could do that? Actually, because if all of them do that, they could quickly become targets of lysis themselves, because the T cells will suppose that these cells are infected actually. So they will get killed by releasing substances or different mechanisms uh, given by these CD8 T cells. Uh, Actually, they need to be licensed by T helper cells. So we will talk about this a little bit later. After we talk about another question, which is how cross-presenting cells handle self-peptide presented on MHC class 1. So as it is shown here, this, for example, this could be self-peptide. So does not this causes a break in tolerance? Because, for example, when it expresses a self-peptide, doesn't it cause break in tolerance? For example, this yellow color are self-peptides. So what is the answer? Actually, no, it doesn't cause break in tolerance, because actually there is a process or a licensing process given by T helper cells. For example, when an antigen presenting cell capture a virus infected cell which processes the virus, it processes it and express both of antigens. So when it engages with a CD4 T cell, the T cell, when it recognizes the self-peptide, it doesn't give any order to the antigen presenting cell actually, but whenever uh, it recognizes the virus derived antigen, it causes the activation of antigen presenting cell later on, which will cause to activate CD8. So this is called licensing. So this license is given by CD4 T cell. The process we just talked about cause uh, cross priming. So it causes the activation of CD8 T cell. So whenever there is a self peptide, actually the antigen presenting cell is not given license to activate any CD8 T cell. So it may lead to tolerance, as it is called cross tolerance. This licensing process has advantage to avoid accidental injection of CD8 T cell to, for example, self peptides. And this is how, for example, when an antigen presenting cell itself is not infected, it could capture, for example, a virus infected cell, or, for example, cancer-derived antigens or cancer-infected cell, and present to CD8 T cell to elicit a response. Until this point, we were talking about MHC molecules, class 1 and class 2. We were talking about displaying or presenting peptide antigens, but actually there is some process which is called presentation of non-peptide antigens. So there is a process to display other antigens other than peptides. So let's see, how does this happen and what molecule do this process? Many records or many reports now indicate that various types of T cells, such as the T cell expressing gamma and delta, as well as alpha and beta T cell receptors, can react against lipid antigens, such as mycolic acid from Mycobacterium tuberculosis. These antigens are presented by members of CD1 family of non-classical MHC molecules. So this is the structure of the CD1. We see deep binding pocket of A, C, and F, and a deep tunnel produced by the T that accommodate lipid antigens in a food and shoe fashion in the
member of CD1 family. So it's a kind of non-classical MHC molecules. Structurally, it is similar to MHC class 1, as we see it's composed just like alpha 1, alpha 2, and alpha 3 domain plus beta 2 microglobulin, but actually functionally it is similar to class 2 as it reacts with exogenous antigens. There is 5 human CD1 genes. All are encoded on a chromosome, separate from classical MHC molecule, and display very limited polymorphism. So much like classical MHC molecules, CD1 proteins are formed by a transmembrane heavy chain and through extracellular alpha domain associated non-covalent with beta-2 microglobulin. However, as we said, it reacts with the exogenous antigen, so it's more similar in function with class 2 MHC molecules. It is also expressed by many types of immune cells, such as thymocytes, B cell, dendritic cell, and also some members of the family have also been found on hepatocyte and epithelial cells. Many different lipids, actually, many different lipid and lipid structures, lipid link structure have been found to associate non-covalently with CD1 molecule. In general, most of these glycolipid and lipoprotein antigens, where the lipid moiety fits into the deep pocket within the CD1 binding groove and the hydrophilic head group remains exposed, which then reacts with the T cell receptor. Uh, some of these pockets correspond roughly with antigen binding pocket in classical MHC class 1 and allow antigen access to a deep groove through a narrow opening. So as we see here, the antigen comes into here as just depicted by this peak. Actually, this is a ribbon diagram of the binding groove or pocket of human CD1B complex with lipid. Compare the class 1 MHC molecule binding peptide antigen. Um, so what is the mechanism of this binding? So what happens during the process is hypothesized that short chain of self-lipid with relatively low affinity are loaded onto CD1 molecule in the rough endoplasmic reticulum when then it is translocated to the membrane. This binding of the low affinity lipid, it is just for assembly process, as we saw in previous processes of endo and endogenous antigen process, which the binding of the clip in the class 2 and the antigen in class 1 was very important process for the assembly. And this is just the same, but in here the lipid takes the role. This self-antigen loaded CD1 molecule then travel to the cell surface, and in some cases exogenous lipid may be exchanged with the low affinity self-antigen, like which with the MHC class 2 molecule after endocytosis and movement to lower pH environments. CD proteins are believed to exchange their self-peptide and then return to the surface. This is how actually the process of loading and exchange with a low affinity lipid antigen happen. There is many kind of cells which react with these cells actually. As we talked about, for example, gamma and delta T cells, as well as there is another kind of cell which is called natural killer T cells. Uh, This kind of T cell are small population of T cell that express antigen receptor protein that are similar but not identical to those CD4 conventional CD4 and CD8 T cell. Actually, uh, the delta and gamma T cell can recognize different types of antigen, including some proteins, lipids, as well as phosphorylated molecule and alkyl amines. These antigens are not displayed by MHC, and gamma and delta T cell are not MHC restrict, so it is not known if a particular cell type or antigen display system is required for presenting these antigens. So now we know this, despite or in addition to expressing lipid antigens in CD1, there is some kind of antigens. Till now, it is not known by which system or which cell kind, cell type they are expressed. For example, as I said, alkylamines and some phosphorylated molecules. Uh, Also, it's not known which cell actually reacts with them, as it was shown that the gamma and delta T cell, it is not MHC restricted. So now here we join the end, sorry, we joined the end of our topic, which was complementary for MHC molecule. This was part 2. I hope you get benefited, and please don't forget to subscribe my YouTube channel, and goodbye.